Hi everyone. In this video, we will discuss pediatric dosage forms tailored dissolution approach. Pediatric dosage form are not just a smaller version of adult medicines. They come with unique challenges driven by children's physiology, growth and development. First, dose flexibility is critical. Children need age and weight appropriate dosing, which requires adaptable formulations like liquids, dispersible tablets, and mini tablets. Second, swallowing ability varies significantly across age groups. Infants, toddlers, and young children cannot swallow standard tablets, so dosage form design becomes a key consideration. Third, taste and palatability are crucial. Unpleasant taste can directly impact compliance, making flavor masking and excipient selection essential. Now, from a dissolution perspective, testing must reflect these specialized conditions. Media composition, volume, and agitation should simulate pediatric gastrointestinal environment, not adult settings. Finally, all pediatric products must comply with strict regulatory expectations. Guidelines emphasize age-appropriate formulations, safe excipients, and biorelevant dissolution methods. In summary, pediatric drug development demands a patient-centric approach where formulation design and dissolution testing go hand in hand to ensure safety, efficacy, and adherence. Children have very different gastrointestinal physiology than adults. They have higher gastric pH, shorter residence time, and much smaller fluid volume in the stomach and intestine. This directly impacts drug dissolution and absorption. Pediatric dosing must be flexible. We often need fractional doses or sprinkle on formulations to match body weight and age. Test masking is essential for compliance, but it can delay dissolution and change drug release behavior. That's why pediatric doses form vary widely, including liquids, oral disintegration tablet, granules, and mini tablets. In short, GI physiology, dosing flexibility, and taste masking must all be considered together when designing pediatric formulations and dissolution methods. Let's look at common pediatric dosage form. Mini tablets 2 to 4 millimeters are small enough for infants to swallow and need size appropriate dissolution testing. Sprinkle capsule and granules are mixed with food, so dissolution must consider food effects. Orally disintegrating tablet. Breakdown in the mouth, requiring rapid disintegration and release testing. Syrup suspension and solutions are liquid forms, so dissolution focuses on drug release and stability. Multiparticulates like coated pellets or beads control drug release, so testing depends on coating thickness and design. In summary, each dosage form needs a customized resolution approach based on size, coating, and intended use. Taste marked particles often use pH triggers or delayed dissolution to hide bitterness. These mechanisms must be properly evaluated in vitro. Taste masking coating slow drug release. So test conditions must capture this effect. Very small particles can float or clump, creating variability in results. For sprinkle products, food effects are critical. Applesauce, yogurt, or milk can change drug release and absorption. Small fill volumes make media scaling difficult, and dose splitting adds more variability. So, robust dissolution design is essential to ensure reliable pediatric performance. When designing dissolution methods, physiological conditions must be considered, especially in early R&D biorelevant studies. Infants have much higher gastric pH, sometimes as high as pH 4 to 6. They also have very low gastric fluid volume, often just 10 to 50 millimeter. Gastric emptying is faster, reducing drug residence time. Enzyme systems are still immature, affecting drug breakdown. And while salt level differ, changing solubility and absorption. That's why pediatric dissolution method must reflect real physiology to generate meaningful data. In low fluid pediatric environments, poorly soluble drugs dissolve even more slowly, directly impacting absorption. For fixed dose combinations, the medium must support all APIs, not just one. If biorelevant is needed, use physiologically relevant pH, typically between 3 and 6. Consider simulated pediatric gastric fluid or SPGF for early R&D studies. Use reduced buffer strength to mimic lower vial salt levels. Avoid strong surfactants unless there is clear justification. And always consider co-administration media like milk or infant formula as they can change drug release. Standard adult solution use 900 ml volume, but this does not reflect pediatric fluid volume. Unfortunately, in QC, we are limited by equipment standardization. USP apparatus 2 is widely used, but it can cause floating or mini particles. USP 1 apparatus works better for mini tablets and beads. Mini vessel system with 100 to 250 milliliter better simulate pediatric conditions. Use apparatus USP4 is ideal for multiparticulates and extended release coated granules. So, apparatus selection is critical for meaningful pediatric dissolution testing. Visual inspection is critical during dissolution testing. Light particles can float or form surface rafts, leading to false results. For ODT and granules, agitation may need to be increased, typically 50 to 75 rpm, to prevent coning. For very small units, use basket or sinkers to keep them submerged. Most importantly, maintain consistent hydrodynamics to avoid variability and ensure reliable data. Test masking coatings commonly use polymer like Utazit E, ethyl cellulose, and HPMC. These coatings delay drug release, so a proper pH trigger is needed. Once the dose is swallowed, polymer thickness directly controls the dissolution rate. But aggressive media cannot destroy coating too early, giving misleading results. So, release must be evaluated both after disintegration and in later gastric stage. It's delicate balance, mask the taste, but release fast enough for efficacy. Regulators expect dissolution testing both with and without food dispersion. Some API interact with milk and fat content, changing drug release. Common test food include applesauce, yogurt, and pudding. The dosage form must stay intact on food, no premature rupture, so drug release must be tested after dispersion on food. And coating should tolerate mild chewing forces and ensure safe pediatric use. For suspensions, dose uniformity is critical. They must show good redispersibility after shaking. Particle setting can bias sampling, so sedimentation must be controlled. Viscosity affect drug release and must be optimized. Finally, check pH and preservative effect, as both can change dissolution behavior. So, stability under agitation is essential for reliable pediatric dosing. In low volume media, there is a high risk of drug absorption, which can distort results. So, robust filter validation and recovery studies are essential. Sampling multiparticulate is challenging. Test masking fragments, can clog filter. Methods must be sensitive and specific for very low pediatric doses and confirm drug stability in small volumes. Finally, method robustness must reflect real pediatric use conditions for reliable data. In summary, pediatric dissolution is a regulatory priority. Product must show reliable performance across ages, foods, and real administration practices. Start by understanding pediatric physiology, select media that reflect child gastric conditions, use the right apparatus, often mini vessel or baskets, account for test masking, food effect, and particle behavior. And finally, validate for low dose accuracy and method robustness. That's how we ensure safe and effective pediatric medicines. Thank you for watching. In next video, we will discuss floating and gastroretentive tablet dissolution method selection. Please subscribe us for getting update on upcoming industry application oriented video and stay tuned because in this 62 video series we will cover dissolution method development, validation, troubleshooting and many real industry examples. Please share your queries in comment section. See you in next video. Thanks.